Welcome to our virtual tour. We are working at Griffith University in Australia. It is in the state of Queensland, and Griffith University has many campuses. We are working at two of them. One is in the capital of Queensland, Brisbane, and the other one is in the Gold Coast. I'm Associate Professor Jenny Ekberg from the Spinal Injury Project here at Griffith University. First, I just wanted to tell you about our approach towards the treatment for spinal cord injury. So what we do is that we look at the needs of the end users. You guys, people who have spinal cord injury, their friends, their families, the entire community, and we interact with you a lot, and we want to see what you need so that we can work backwards from that. And now I want to welcome you into our laboratory here at Griffith University, up here. So please come with me behind the scenes. So before we go into the lab, we're going to start by going into the nose. If you imagine shoving your finger all the way up into your nose, and you couldn't even get all the way up to the top, but at the top is where you find the cells that we are working with and that we're aiming to transplant from a person's nose into the spinal cord. Because all the way up the nose, you find a nerve that mediates the sense of smell. It's called the olfactory nerve, and it's very unusual because it's constantly regenerating and renewing itself throughout a person's life. And that doesn't happen anywhere else with any other nerve in the nervous system. And the reason why this nerve is regenerating so well is because it has a special type of cell in it. And that cell is called olfactory sheathing cell. These are the cells that we are working with, that we're taking from the nose, transplanting into the spinal cord. So the first clinical trial in humans using these cells were actually performed here on the Gold Coast by Professor Alan Mackay-Sim, who was the Australian of the Year in 2017. His trial showed that it's safe, it's feasible, and what we are doing is we're trying to make it better. So now we're in the tissue culture room, and here is where we grow our cells. We culture our cells in these hoods to keep them safe from bacteria, from anything that can damage the cells. And here we see Suptik and Lynn working in the background, growing their cells, growing the olfactory sheathing cells. While we are here in the tissue culture room, we're going to hear from Dr. Anu Chako more about how olfactory sheathing cells behave.
My name is Dr. Anu Chako and I'm a research fellow. Our team here focuses on understanding and improving the therapeutic potential of olfactory and sheathing cells for better spinal cord repair. Our team was the first one to discover and characterize a very novel function of olfactory and sheathing cells, which is their ability to degrade and engulf cellular debris. Here in this video, the olfactory and sheathing cells, which are the glial cells, which are in red here, are able to move around and degrade the cellular debris, almost acting like a vacuum cleaner, cleaning up the injury site. Traumatic spinal cord injury causes extensive cell death. Cleaning up this hostile injured site is the key for better regeneration. If we can increase the cleaning capacity of olfactory and sheathing cells and control them in such a way that they clean it efficiently, that would improve the therapeutic potential of these cells. Our team here focuses on understanding the cellular and molecular mechanism which controls this cleaning capacity of olfactory and sheathing cells. We have also identified novel compounds which can activate the cleaning capacity of these cells. Thus, the outcomes of this project will not only improve the cleaning capacity of olfactory and sheathing cells, but will improve the regenerative potential of the spinal cord and will thus improve the clinical outcomes.
Now we are going to our other campus, which is just outside Brisbane. We're going to meet Dr. Mariam Murtaza, who's going to tell you more about the work that we do at this campus.
Hello everyone, I'm Dr. Mariam Murtaza, You've heard what olfactory ensheathing cells can do to clean up the injury site. Today, we're standing outside the Griffith Institute for Drug Discovery in Brisbane. Please follow me in the lab to see where we source the cells from and how we can process the biopsies. Olfactory ensheathing cells exhibit strong neural repair properties and can be obtained from the nasal tissue by a relatively non-invasive tissue harvest. When a participant consents to providing a nasal biopsy, a small piece of tissue from the lining of the nose is removed by an ENT surgeon. This is done under visual guidance using a nasal endoscope to identify and collect tissue from a region with high yield of OECs. This tissue is immediately placed in cell growth medium and is transported on ice to the Griffith Institute for Drug Discovery. This sample is processed in a biological safety cabinet under a dissection microscope to remove blood and contaminating bone fragments. Clean tissue is then enzymatically digested and finely chopped to isolate the cells. The expanded cells are grown in large numbers and purified, so they are in a format ready for transplantation into the injury site. To identify different cells obtained from the biopsy, we label them with specific antibodies. This is an example showing cells of interest in red and the other contaminating cells in magenta and green. We purify the cells using different approaches to eliminate the other cells and enrich for the olfactory glial cells of interest. So using a combination of cell type specific markers, we can identify different subpopulation of cells. We can then observe the shape of the cells, determine how fast they can move, and even how they can interact with neurons, and all of this is in a dish. We can also make three-dimensional constructs of cells to investigate if it favors better neural repair properties of olfactory ensheathing cells. And we can monitor the cells in real time to understand them better and to identify cells of the highest therapeutic potential. We are now focused on large-scale manufacturing approaches using bioreactors to produce clinical-grade OECs with the goal of improving our therapy.
So now we're going back to the Gold Coast campus again to find out more about what we do at this location. Okay, so here we are in our main lab. You see one of our scientists, Ali, is working here in the background. So this is where we do most of our work.
I'm Dr. Mo Chen. I'm the one making the 3D culture in our team. And I can make the 3D culture like nerve bridge. It's all formed by cells. I can make our OEC nerve bridge. It's all formed by OEC cells. And we also can make a personalized nerve bridge. We can scanning the damaged neuron and making the suitable shape of our 3D culture. And overnight, you can get a nerve bridge.
My name is Dr. Tanja Eindhoff. To date, our team has established various robust spinal cord and peripheral nerve injury models. At any time, we're working closely with our clinical colleagues to transition something which has been created on the lab bench in a research setting to the everyday application in a hospital. We are able to highly customize and individualize our bioproducts like the nerve bridge by using the patient's own cells and tailoring the actual nerve to the specific injury site while maintaining at the same time ease of handling and user-friendliness for the surgeon in the theater. Over the course of many months, we are collecting various parameters from our projects. The analysis and assessment takes time, patience, and perseverance. To give you an example of our capabilities, we used the chicken sciatic nerve and imaged it and created a 3D printed exact model of the nerve. We then created a mold and filled it with our cell treatment to recreate the same shape the initial nerve had and now created a nerve bridge. The challenges we're working at the moment with are the methods and ways how to fixate the nerve bridge within the patient's body. We are therefore using various scaffolds and different bioglues. So far, we have made much progress, and I'm very excited and enthusiastic to be part of this great team.
Now we're in the microscope room. Here we have very advanced microscopes, excellent for studying the olfactory and sheathing cells. And we're going to show you how these work in detail.
Oh hi, I'm Dr. Todd Shelper, and I'm part of the team here at Griffith University looking to develop cell therapy to treat spinal cord injury. I spend most of my time looking down one of these microscopes. And what we do here is to try to see what's happening in the spinal cord injury on a cellular level. What we can look at is processing all the tissues of spinal cord injury, and this involves quite a huge process. We have a large team that helps prepare the tissue and place onto glass slides, and this allows us to use these microscopes to see what's happening in a cellular level. This box of slides just shows you how thin the sections have to be able to use the microscope to see what's happening. So this image on the screen is an example of the type of images we can take on our microscopes. Here you can see the two stumps of an injured spinal cord with the non-functional spinal material in between. The spinal cords here are coloured in red, and what we're able to achieve with our cell therapy is transplanting the OECs into this injured spinal cord. So with the amazing instruments that we have here, we can image down to a cellular level and see exactly what's going on with our transplanted cells here in yellow and the injured spinal cord. We can also see in 3 dimensions what's happening. We can zoom in, look at how those individual OEC projections are interacting with host neurons. And this result is exactly what we're looking for. This gives us real confidence that the results we're seeing here will translate into human trials. So these amazing images are made possible by instruments such as the microscope right here, which was funded by the Perry Cross Spinal Research Foundation. This allows us to get images much quicker and a much higher resolution. Not only the spinal cord work, but we can also look at peripheral nerve work. This is an example of the peripheral nerve work we're doing. And as you can see in this picture, in red here are the nerve cells, highlighted in red, so this is a sciatic nerve, with our nerve bridge running down the sides. So this allows us to zoom in on a single cell level and see how the nerves are interacting. Thanks for coming along and spending some time with me in the microscopy room. I'm part of a great team that works together to achieve these results, and I can't wait to see what happens in the future.
So now that we've seen the microscopes, we're going to go upstairs and meet Dr. Marie-Laure Vial and Dr. Andrew Rayfield, and they're going to tell you more about our rehabilitation program.
Hi everyone, I'm Dr. Marie-Laure Vial. You've just heard about all the exciting work we have been doing in our lab, but cell transplantation alone is not enough. The nerve cells must be stimulated in order to find the right connections. This can be achieved with intensive long-term rehabilitation. So previous clinical trials around the world have shown that the inclusion of a rehabilitation program post-cell transplantation has led to successful outcomes. Very exciting news, we are now starting a rehabilitation clinical trial. This trial has been co-designed with the community. We have established the Spinal Injury Project Consumer Research Panel. We have received many enthusiastic applications, and we have chosen 10 people living with spinal cord injury from around Australia to be part of this panel. We are in discussion with the consumer group. They help us conduct more focused and translatable research and help us ensure that we are developing the best trial possible.
My name is Dr. Andrew Rayfield. We're undertaking a physical rehabilitation trial. This is to work out what works and doesn't work in preparation for a future cell therapy trial. We're going to undertake this physical rehabilitation trial here on the Gold Coast. And what better place to do it? With our base here at Griffith University, just across the road from the Gold Coast University Hospital. We're also part of the Gold Coast Health and Knowledge Precinct, and we're just down the road from Burleigh, where Making Strides is located. For the rehabilitation, we'll need at least 15 people to undertake rehabilitation for 2 hours a day, 5 days a week, for 16 weeks at Making Strides on the Gold Coast. Then those participants will return to their home communities and undertake a further 10 weeks of rehabilitation with the aid of online support. We'll be following our participants in their rehabilitation journey to see what works and doesn't work. We need this information to improve the design of the rehabilitation program in order to meet the needs of the participants. This will allow us to maximise our chances of success for the full cell transplantation trial.
So now that we've heard about our rehabilitation program, I'm going to introduce you to Associate Professor James St. John, who's going to tell you more about this program and also about our plans for future clinical trials.
Hi, I'm Associate Professor James St. John. We've just finished the main lab tour and now we're going to take you to Making Strides at Burleigh. It's just down there a few kilometres. So let's go. Hi everyone, here we are at Making Strides at Burleigh on the Gold Coast. This is the place where we're going to be doing the rehab feasibility trial. It's exciting to be here to see how everything operates and to see the journey of the participants. We have to get this journey right so that we can get the best possible chance of success for the full cell transplantation clinical trial. I'm going to introduce you now to Jim Barrett, who's one of the bosses here at Making Strides, and he can talk to you about what makes Making Strides so fantastic.
Hey guys, I'm Jim Barrett. I'm one of the crew members here at Making Strides. Making Strides is a specialist neuro centre. We've been around for about 7 years now. We work predominantly with spinal cord injuries, but all different neurodisabilities at the same time. A lot of our crew members have sort of been here from the start and got at least 5, 6, 7 years neuro experience, which is really cool because it allows us to really specialise on the individual, what their current function is, and then match that with what their goal is. So because we've been around for a little while now, we've got some really cool specialised equipment specifically for this population, and whether we've customised it and made it ourselves or whether we've sought it from all around the world, some of the red cord stuff you can see behind here. And we've also now partnered really well with different allied health professionals. We've got orthotists on staff, massage therapists. We work really closely with physios who are here at the moment, as well as we've got in-house, we've got some specialist nursing guys who are gurus with wound care and bowel and bladder and all the stuff that people need while they're training and living lives with a spinal cord injury. So we're stoked to be part of the Griffith stem cell study and really excited for what might happen there, and keen to see what sort of results we can do when we add the training modalities with the science breakthroughs that everyone's getting.
Hi there, my name's James Harvage and I'm the on-site orthotist here at Making Strides. I actually work for Neuromuscular Orthotics. Our head office is based in Melbourne. We've had a partnership here with Making Strides since about 2015. Basically, our mission is to help support people with a neurological condition, whether it be a spinal cord injury or MS, to help reach their goal. Sometimes that can include walking, it can include other tasks, such as cooking, cleaning, whatever it might be. We do that with different types of supports, commonly known as orthoses, to help facilitate normal function, again, in line with whatever their goal might be. We're just getting them back to doing those things, and we're helping them in whatever way that we can here with the team at Making Strides.
Hi everyone, now we're back here at Griffith University and I'm going to go through a summary of what you've seen before upstairs and also where we hope to go into the future. What we haven't talked about yet today is the incidence of spinal cord injury. There's about 15,000 people living with spinal cord injury in Australia right now. And every year there's about another 300 people added to that list. Half of those people have high-level quadriplegia injuries and the other half have paraplegia injuries. There is no current cure, but we're working very hard to make that happen. It might seem strange, taking cells from the nose to repair the spinal cord, but it seems to be working. And it's not just us who've shown that it's working. It's been shown before by Alan Mackay-Sim, but also by people overseas. This guy here is Derek Figia. He'd been stabbed in the back, complete spinal cord injuries, and this Polish surgeons, along with the researchers from University College London, Professor Jeffrey Reisman, transplanted cells from his nose into the spinal cord. He regained motor activity and also feeling. It wasn't easy, he had to do high intensity rehabilitation to achieve this. But this shows that taking cells from the nose and putting it into the spinal cord can work. but we have to make the therapy better. And this is where we come in, our research in the lab. You've seen upstairs Mo Chen, great bioengineer who can make nerve bridges. Well, we've been focusing on creating that pipeline so we can make it suitable for use in the clinic. We've now got the timeline down to about 36 hours. We can image the patient's injury site, find out what shape that injury site is so we can make the nerve bridges. We can then 3D print the templates that we need to make those nerve bridges to suit that injury site. Then once we have the cells ready, we can add them to that template to make the nerve bridges. In this case, it's a peripheral nerve bridge, but we can do the same thing for the spinal cord injury nerve bridges. And then after only 36 hours from the time of imaging to having that nerve bridge, these nerve bridges are ready for transplantation back into the patient. So we're very much focused on making it translational, thinking about what the clinicians want, thinking about what the patients want. And what is so exciting, when we transplant the cells into the spinal cord, we get great results. These red cells here are the cells that we transplant, these olfactory and sheathing cells that come from the nose. And once they're inside the spinal cord, they have the right morphology, the right shape, the right length, the right branches, and they interact with the white nerve cells beautifully. When we go to high magnification view, we can see that these white nerve cells are wrapped up by the red olfactory and sheathing cells. And that's what they're supposed to do, ensheathe. They ensheathe the nerve cells to protect them and guide them across the injury site. So these results here are really exciting, and it correlates with improvements in functional and sensory recovery that we see. So the question is, how do we get from our lab research to getting it into the clinic? This is our plan for a clinical trial. We're focusing on chronic injuries because that's the safest approach at this point. We do think our therapy is suitable for acute spinal cord injury, but we're focusing on chronic for the moment. So someone with a chronic injury will have to undergo a period of activity-based therapy, physiotherapy, exercise physiology for some time. Then we can take cells from the person's nose, purify them, make them into a nerve bridge, and transplant them back into the patient. That's the easy bit. Then they have to keep on doing long-term intensive rehabilitation. And that might take months and years to get the best possible chance for recovery. And that's why we have to test the rehabilitation program. It's the real tough bit to get it to work. And so that's why we're so excited that we can now progress to the feasibility study for testing the long-term intensive rehabilitation. It's going to tell us what works and what doesn't work. And with our partners at Making Strides down in Burleigh, you can see how enthusiastic they are and how comprehensive they approach each client and the journey. We hope that we can get the best possible outcomes. And so how do we get to a clinical trial? Well, we also continue to have our ongoing research here in the labs. We're getting the approvals for the clinical trials underway. We've started the rehabilitation trial. We still need more funding to get enough people so we can test it properly. But then ultimately, we want to progress to the cell transplantation clinical trial. It's going to cost a lot of money, but that's what we need to get this thing to work. And when you think about how much it costs compared to the total socio-economic cost to Australia, it's only one day's cost. So it's worth it. It's worth it from a socio-economic point of view. It's worth it from the person point of view. And together, we can do this as a community. We can't do it on our own. We need your help. And it's the help of many people. It involves the community, people such as yourselves, people living with spinal cord injury, their family, their friends, and everyone else. The local government to provide services that we can access these facilities and rehabilitation in our local areas. State government has to be involved, federal government, then also our industry partners, such as you saw at Making Strides, but also with assistive devices. The clinicians, critically important so that we can get a therapy and cell transplantation that works for what they need to treat the patients. And of course, we need funding through philanthropy, through state government agencies as well, and through donations. But together, we can get this cell transplantation working. We've got many different stakeholders, particularly the Motor Accident Insurance Commission of Queensland. We're very thankful for their support, Clem Jones Foundation, and of course the Pericross Spinal Research Foundation. So you've heard from quite a few of our people today, but that's not all of us. We have a big team here at Griffith University, more than 30 people from all around the world. We have something like 16 different countries represented here at Griffith University in our lab in the Spinal Injury Project team. 50% male, 50% female. Lots of different experiences. Diversity is our key because we need new ideas to help us get that next extra bit to make the therapy even better. So we're sorry you can't be here with us today because of COVID-19, but I hope you've enjoyed our tour. And we look forward to continue sharing our journey with you in the future, either in person or virtually. And we'd love to have you visit us here once COVID restrictions are gone. So I hope you enjoyed the tour, but just before we go, we've got one more thing to show you. We work and live on the Gold Coast, so what better place to enjoy ourselves but on the beach? So let's go to the beach. Hi everyone, as you can see I've swapped my laptop for a beach towel. What a great place it is to do research here on the Gold Coast. Our team is totally committed to making this therapy work. We want people to not only to be able to walk along the beach, but to feel the sand between their toes. We want this therapy to work, to restore motor, sensory and autonomic function. Together we can do this, and the possibilities are limitless.